how do you start to work with his process to develop iPS cells?
Well, the problem was Shinya Yamanaka originally published that he could do it in mouse cells, not in human cells.
Yeah.
And there was a huge race on.
Mouse embryo cells.
It was mouse fibroblast adult cells that he reprogrammed back in time to a state like an embryonic stem cell. But he hadn't done it in human cells. And there was a huge race from between 1996 and 1999 to do it in human cells. And in fact, Jamie Thompson published simultaneously with Yamanaka showing that they could do it in human cells. So it was a dual publication, and once we did it with human cells, that's when everything opened up. So now we can make these powerful cells, which are just like embryonic cells from human cells. And I think that was a switching point for many, many people, including companies like Johnson & Johnson, who refused to use embryos, now suddenly could use this new cell type. And the process is remarkably reproducible. You know, sometimes you get a discovery in science in a big impact, and you never hear from it again. How many times have you done a news article on something that disappears? Cancer cured, you know, and then a week later it's found that it couldn't be reproduced. So these cells were reproduced in hundreds of labs within a year or two. So that this technology is straightforward, every lab is reproducing it. And it's the most remarkable thing that every cell in our body, the DNA of the cell is identical pretty much. It's just everything around it that's different. So the reason you have a hair cell versus a brain cell versus a skin cell is not because of the DNA, it's because of what we call the RNA and the protein, because the protein is what shapes the cell. And so once we understood that, and Dolly the sheep proved that you can clone an adult, the DNA is the same. That means that if you can manipulate the DNA, you can morph it into anything you want. And that was really what Shinya Yamanaka showed. So just fascinating science. And of course, I'll talk about how that impacted me. I was with Jamie Thompson, so I found out about this 6 months before anybody else, because he and I shared information. And then we put a nice paper together for Nature showing that we could use this technology. Everybody was thinking, well, we can transplant patients with cells. But we actually set a whole new area up called disease modeling. We call disease in a dish.
Yeah.
Which means
in a petri dish.
So for complex diseases, you can actually take the cells. So if you had a rare disease, you could take your cells, take them back in time, and then if it was a brain disease, I can push them forward to be neurons, and I can look at the neurons that were in your disease in the dish.
I would love for you to, Clive, to try to translate this to a clinical state. Like you said, disease in a dish. You could then take medication or chemotherapies or whatever to do the work on the dish and not on the patient.
Correct. So say, best example is if you had a heart arrhythmia, which means your heart doesn't beat correctly. If I took your cells, reprogram them, I could make them into beating heart cells, and in my talk today I'll show some. Those beating heart cells will reflect your arrhythmia. So if you wanted to try and treat it, you could put drugs onto cells in the dish instead of on you, and I could try 1,000 drugs and find the one that works for you, for Jorge, not for everybody. So yeah, but we call it personalized medicine. It's happening in genomics and cancer right now for cancer therapy, and these cells will, on a clinical setting, at Cedar Sinai, our goal is to have these cells produced from every patient.
Correct.
And so when you come to the hospital at Cedars, you will have your cells in a dish like an avatar, and the doctor will say, right, it's going to take a week and I'll find you the right drug.
I think this is a fascinating part of what your work is being for the future.
I think it's something that the public doesn't quite get, because I think when they think regenerative medicine and stem cells, they think transplants, cells are going to cure me. I think the biggest impact of this technology is going to be on drug treatment and therapy, personalized drug treatment and therapy, where we have your cells in a dish, whether it's the bone cells, if you have a bone disease, brain cells, heart cells, we can grow all of these cell types.
What kind of hope can you give to ALS patients?
So a big focus the institute I run works on all different diseases. My lab in particular works on ALS, or Lou Gehrig's disease. And we are actually attacking this using regenerative medicine at Cedar Sinai from two different ways. One is we're taking patients with ALS, and just as I was discussing previously, we're putting their cells in the dish and then turning them into the neurons that die. In the case of ALS, it's neurons that connect to the muscle. So in ALS, you get paralyzed because the neurons down here in your spinal cord die. We don't understand why. If we could understand why they die, we may have some clue to develop a therapy. And until you understand the mechanism of why they die. So we've taken patients with ALS, taken their cells, made them into these powerful pluripotent cells, and then turned them into the motor neurons that die in the disease. And now we're trying to understand the reason they die, and then because we can see them dying in the dish, we can give drugs to those cells, just like I was discussing for the heart. So that's one area that's really progressing rapidly at Cedar Sinai and around the world. So that's called basically modeling ALS in the dish. 10% of patients have some kind of gene associated with the disease, but 90% of patients don't have any family history, and they're called sporadic. We haven't yet found any gene mechanism. It's the same for Parkinson's. 10% of Parkinson's patients is genetic, 90% sporadic. So it's interesting. Alzheimer's, 7% genetic, 93% sporadic, i.e. no known gene. So all of these neurological diseases are probably what we call complex genetics. So we're looking at the whole genetics of the cell in ALS and working out what cloud of genes or proteins might cause ALS as well. So here we now can generate your own cells. What's interesting is when you make these induced pluripotent stem cells, these embryonic stem cells from your own tissue, they're rejuvenated. You take them from being 70 years old to 0 again. So what we're trying to do is then make them into neural tissue and put them back in the brain to rejuvenate brain areas so that the aging process is reduced. And essentially that's what we're doing in ALS. We have a trial now that will start with 18 patients at Cedar Sinai in November this year, with any luck. We've still got to get through the FDA, but we're looking quite good. And that trial will be the first really to try putting these cells in, and we're actually combining this with something called gene therapy, where we modify the stem cells to release a powerful drug that you can't get into the brain. So it's a combined therapy for ALS, and we're very excited about this. And if it works for ALS, and we slow down disease progression, we can then use it for Parkinson's disease, so there's a big interest there with Parkinson's and Alzheimer's to slow down. And it's really putting in these new support cells in the brain that get old, and it seems like some of the mechanisms of aging are actually responsible for a lot of these neurodegenerative diseases. You don't get Alzheimer's when you're 25, and so it's a complex problem with aging. So we found now from iPS cells we can create new blood cells.
Yeah.
So what we're doing is with mice at the moment, we hope with humans, is rejuvenate the blood system by, you do irradiation, the bone marrow where all your blood is made, you put the iPS blood cells back in, and now you have a young blood system because the cells integrate and exchange the old cells for new ones. So it's like going for an oil change. Correct. You have a new, and we found these mice, unbelievably, their brain capacity is normalized. Their activity goes back up to normal. So we have a mouse that's 300 days old that's acting like 100 days old. From my perspective, what we need to do is slow down the aging process, and that will simultaneously wipe out Alzheimer's, ALS, probably Parkinson's, and Huntington's disease. Yeah, my goal is for a health. We talked about had a symposium on what we call healthy aging, and in 100 years, you no longer will. It's just like polio is being cured. We'll cure aging, then we'll have a social problem of how long should we allow people to live. It's interesting. Cancer is caused by a lack of killing dividing cells. Our body has cells that divide all the time. We have mini cancers forming in our body in healthy people all of your life. The reason we don't get cancer is the immune system detects those dividing cells and kills them. So, if you have a healthy immune system, you will kill cancer cells.